Welcome to Chapter 5, the structure and function of large biological molecules, also known as carbon-based molecules, also known as macromolecules. We're going to be breaking this up into a few different presentations. This one's going to talk about monomers and polymers, and then carbohydrates. The macromolecules, there's four of them. There's proteins, carbohydrates, lipids, and nucleic acids. As this flow chart shows, that each is broken down into smaller things, has different properties, but they all have monomers and polymers. Monomers are the individual units or building blocks that can combine to form more complex molecules called polymers. And if we look here, we have three individual things linked together, forming a short polymer. And we have another one that we're going to try to tack on. This would be a monomer, and when we do that, we form a longer polymer. Once again, those polymers would be the macromolecules, so the proteins, carbohydrates, lipids, and nucleic acids. Now, how we link the monomers together is a process called dehydration synthesis or dehydration reactions. So dehydration, removal of water. When the HO and H groups combine, they form water, two H's and an oxygen, and that is removed, resulting in a bond. Now, these reactions can go in the reverse as well. We can take a polymer with the bonds, add in water, and then separate those bonds again. That would be hydrolysis. Dehydration and hydrolysis are both facilitated by enzymes, which reduce the amount of activation energy needed for the process to occur. An example of where these processes occur in our body is in our digestive tract. We ingest the macromolecules, well, three of them anyways, when we eat food, and they undergo dehydration in the stomach. This allows the polymers from food to be broken down into smaller subunits, or the monomers, that are capable of getting into our bloodstream. And once these monomers are dispersed to the cells of the body, hydrolysis occurs to reassemble the polymers based on the needs of whatever cell they happen to be delivered to. There are three types of carbohydrates that we're going to be discussing in this course: monosaccharides, disaccharides, and polysaccharides. Notice they all have saccharides and a changed prefix, mono 1, di 2, poly many. The first group, the monosaccharides, are usually in combinations of CH2O. Notice that the ratio of carbon to hydrogen to oxygen is a 1 to 2 to 1 ratio. That is usually the case with all monosaccharides. Glucose, the most common one looking at it, follows that principle. For every carbon, there are two hydrogens, and for every oxygen, there are two hydrogens. So C6H12O6, or glucose, follows that pattern. Monosaccharides include carbonyl groups, which we discussed in Chapter 4, which would be a CO group in the carbon skeleton. And if you recall, there were two types of carbonyl groups, the aldoses and ketoses, or also called the aldehydes and ketoses. Once again, the difference is the location of the carbonyl group, whether it's at the end of the carbon skeleton or embedded in the middle of the chain. You'll also notice that the aldehydes and ketoses have a couple different formats: the trioses, pentoses, and hexoses. And generally, they follow that 1 to 2 to 1 ratio we were talking about. The hexoses do 6, 12, 6. The trioses do 3, 6, 3. The pentoses, not quite, so it's a general rule. One last thing that I would like to point out is sometimes, like C6H12O6, that's could be different sugars in this case entirely. This one's glucose, this one's galactose, same molecular formula. Notice here the switching of the H and OH groups from one to the next. Looking at glucose, once again, our big one here, especially in cellular biology. It can exist in linear or ring forms, and if you notice the double arrow for the reaction, it can go either way. This is it as a chain, and this shows it forming a ring. The ring structure of glucose off to the right hand side is what we see with glucose in solution. Doesn't take the linear format in solution. The ring is what you see. And looking at this one off to the right, we see that the carbons have been numbered as carbon 1, carbon 2, carbon 3, 4, 5, and 6. That's to denote the location of the carbon, and so that we know what carbon is being spoken of during certain bonds. We see those numbers of the carbons used in the bond that formed right here. This is called a 1,4 glycosidic linkage. A glycosidic linkage occurs between two monosaccharides in the formation of a disaccharide using the process of dehydration. So in this case we have glucose and glucose forming a glycosidic linkage between the first and fourth carbon of these two monosaccharides as these react with one another to dehydrate. The more complex carbohydrate is a polysaccharide made up of many monomers, and it has two primary functions. One, storage, and the second is structure. Looking at storage, we store energy for plants and animals using polysaccharides. In plants they use starch. Starch is their polysaccharide, which can be broken apart into monomers and then used for energy later on. And for animals, it's glycogen, which can be broken down into the individual glucose molecules for energy. In plants, it's typically stored in the chloroplast. We can see starch granules right here. And in animals, it's usually in muscle cells or liver cells. And we see some of that floating around in the cytoplasm right here. Looking at the structural function of polysaccharides, we have to first take note that glucose can take on one of two different ring structures, either the alpha or the beta. Notice the location of the hydroxyl group, which is the OH, at the end. This creates different glycosidic linkages to occur, resulting in slightly different polymer structure, as we see down the bottom here with starch and cellulose. Polymers with the alpha glucose are more helical, which is perfect for storage as they can kind of coil up a little bit more. However, polymers with the beta tend to be more straight, which reinforces strength, especially in the cell walls of plants. Here we can see those cell walls and the beta glucose molecules forming cellulose. As you can see, they arrange themselves kind of in parallel rows. We can see that these two components of glucose are bonding with one another. These two are bonding with one another, and these ones are bonding. So they're all bonding with one another. So not only do we have strength going linearly, but also perpendicularly with the cellulose strands as well. So once again, beta-glucose molecules are great for reinforcing structure and strength, especially in plants. Moving into this slide, we have to recall on knowledge regarding structure and function and how different structures will result in how something functions slightly differently, as we saw with starch and cellulose. Starch is for storage and cellulose more for structure. Well, with that, even though they're similar in appearance, we have to understand that because they are different, we need different enzymes to break apart those glycosidic linkages. And the enzyme that hydrolyzes the glycosidic linkage in starch is not the same enzyme that's needed to hydrolyze the glycosidic linkages in cellulose. Some organisms can break down cellulose. However, humans do not have that enzyme, which is why cellulose is what we call fiber in humans. It's used as fiber to kind of keep us regular, if you will. However, cows, on the other hand, they have bacteria that can break down cellulose. So cows, as you can see in this picture, they graze on grass, and the bacteria in their guts can break apart that cellulose. So cows can actually harness energy from that cellulose, whereas humans can't. And the last polysaccharide that we're going to talk about is chitin, found in the exoskeletons of insects. Once again, we can see the glucose molecule right here with an additional group tacked onto it. Chitin is structural, obviously, as the exoskeleton, but it also can be found in the walls of fungi, and also can be used for our purposes, surgery, as you can see in the picture there.